chapter number 5, the principle of inheritance and the variations. In that we have mostly discussed regarding the monohybrid experiment or the inheritance of a single gene. Even we have also seen a few exceptions like in case of incomplete dominance and the codominance. Now today we have to start with inheritance of two genes. Okay, so let's start with an inheritance of two genes. Inheritance of two genes. Clear? Inheritance of two genes, it can also be called as dihybrid experiment. What it is called as? Dihybrid experiment. In a monohybrid experiment or inheritance of a single gene, we have discussed only regarding one character, the one character that is a height, and there are two different alleles of a height. Capital T for tallness and the small t is for dwarfness. Here is an inheritance of a two gene, means we will discuss the two characters, how these two characters are going to be transferred and what are the rules, what are the ratios they are going to be transferred, and which rule has been framed from this inheritance of a two genes that we will discuss. From a monohybrid experiment or the inheritance of a single gene, the Mendel has developed a two law of dominance, law of segregation. The law of dominance says that the dominant character will express in a dissimilar pair, and the factors or the genes, what we are calling currently, they occur in a pair. In a dissimilar pair, the dominant will express, the recessive will not express when the dominant is present, and the recessive will only express when the dominant is absent. So this is what law of dominance. Even we have also discussed regarding the law of segregation. That means if you see the F1 generation of monohybrid experiment, you can find the genotype capital T small t. So here the characters are not blended or doesn't get mixed, they segregate, okay, they separate while the gamete formation, and hence even though in the F1 generation there is no presence of a dwarf plant, you can find the dwarf or the short plant in the F2 generation. That is possible. That means the Mendel has proposed these two law of dominance and law of segregation from the monohybrid experiment. Similarly, the Mendel has also suggested a law from the inheritance of a two gene or the dihybrid experiment. But first of all, we will learn the dihybrid experiment. That means here the two characters have been considered. The first character that has been considered is a seed shape. Okay, and the second one is seed color. There are two characteristics have been considered. It's an inheritance of a two genes, so there are two characters have been considered. There are two alleles. First is called as round, okay, and the second one is wrinkle. Okay, the round that is characterized as capital R and the wrinkle that is characterized as small r. Now you know that the capital letter is the dominant one. So from round and the wrinkle, these are the two different alleles. Okay, the round is the dominant one, the wrinkle is the recessive one. Okay, now the another character is a seed color. Okay, and that is yellow, and the second allele is a green color. So here the yellow is a dominant, which is characterized as capital Y, and the green that is characterized as small y, it is a recessive character what you can find in it. Okay, now what the Mendel has done, Mendel has took the plant with two characteristics. The first plant or as a parent generation, what the Mendel has done, in the parent generation, Mendel took such kind of a plant. Okay, remember R, Y. You already know that the factors or the genes that occurs in a pair. So this is a pair. This is also a pair. But here we are considering the two character. So here the two genes. One is the seed shape and the second one is a seed color. So this is first RRYY. This is a one parent. He crossed this parent with a characteristic like recessive parent, that is small r, small y, small y. Okay, so what is meaning of capital R, capital Y? This pea plant is round and yellow. Okay, the R that denotes a round, Y that denotes for a yellow. Okay, so here there are two characteristics. Okay, that's why we are calling it as inheritance of a two gene, or it can also be called as dihybrid experiment. Here it's opposite. Small r is denoting from the wrinkle. That means this seed has a wrinkled surface and green. Okay, green. Okay, so this is a round and yellow. This is a wrinkled and a green. Okay, he took this as a parent. Assume that one of them is a female, another one is a male. Okay, clear? One is a female and one is a male. Now what we have to do? We have to form first gametes. So which are the gametes of this? Okay, listen how the gametes are formed over here. First gamete is capital R, capital Y. Remember how the gametes are formed. Capital R, capital Y. This is the first gamete. Okay. Second gamete over here is capital R, small y, cross capital R, small y. I am showing you how to form a gamete. Another possibility, capital R, capital Y, capital R, capital Y. Okay, this is the third gamete. Capital R, capital Y. Fourth is capital R, capital Y. This is the fourth gamete which is possible. Capital R, capital Y. Clear? So I again show you how to form a gamete. First, capital R, capital Y, these two. In the second case, capital R, capital Y, going across. Third possibility, now take this R, capital R, capital Y. This is the third one. Now fourth one, capital R, capital Y. This is the fourth one. This is the gamete of this plant, round and yellow. Now he crossed it with this. Now we can also form its gamete. So what are the gametes of it? See, same thing. Small r, small y. So the first gamete over here is small r, small y. Another gamete going across, small r, small y. So this is the second gamete. Small r, small y. Now the third, like this, small r, small y. This is the third. Small r, small y. This is the fourth. Small r, small y. So here also four types of gametes are produced. Capital R, capital Y, they are the gametes of this plant, round and yellow, and small r, small y, they are the gametes of this plant, wrinkled and green. See here, now you have to do the same thing. You have to do 4 into 4. So how many cross you have to make, or how many square you have to make? 16. 4 4s are 16 square you have to make. Okay, but for easy understanding, we do not go in that much detail. In the F2 generation, we will make the 16, but in the F1 generation, we will make only one, because the reason is here all the gametes are same. So here it's a capital R, capital Y. બધાની જગ્યાએ એક જ representative લઈ લઉં છું. Okay, here also all the gametes are same. Here you can see all the gametes are same. If there are different gametes, you cannot do like this. But in the dihybrid experiment, all here gametes are same. So I am taking just a one as a representative. And here also all the gametes are same. So I am taking just a representative. Okay. Now both gets fused. These gametes get fused. And now you get the first generation. What we call it as F1 generation. Or you can say that filial generation 1. In the filial generation 1, which kind of a plant you obtain? Now mix, fuse together. So you will obtain a plant like this. Capital R, small r, capital Y, small y. Okay, such kind of a plant you will obtain. Now write down its phenotype characteristic. This is the genotype of F1 generation. Okay. Now what is the phenotype of this? Here, from capital R and small r. Which is a dominant one? The capital later. That is a capital R. So here you know the meaning of capital R. It's a round. And here from capital Y and small y. Which one is dominant? Capital Y. And the meaning of capital Y is yellow. So the characteristic of this seed is, what is the characteristic? This seed is round and yellow. This is the characteristic. So all the seeds you can have a possibility, 16 possibility. Okay, તમે કોઈ બી જોઈ લેજો. આ લઈ લો, પહેલું આ લઈ લો, ફર્સ્ટ સેકન્ડ આ લઈ લો, ફર્સ્ટ થર્ડ લઈ લો, ફોર્થ લઈ લો, અથવા સેકન્ડ ની જોડે 1 લઈ લો, સેકન્ડ ની જોડે સેકન્ડ લઈ લો, સેકન્ડ ની જોડે થર્ડ લઈ લો, સેકન્ડ ની જોડે ફોર્થ. સિમિલરલી થર્ડ ની જોડે ફર્સ્ટ, થર્ડ ની જોડે સેકન્ડ, થર્ડ ની જોડે થર્ડ, થર્ડ ની જોડે ફોર્થ. All in all the cases, even same with 4 ની જોડે first, in all the cases you will get round and yellow seeds. Okay, there are total 16 possibility, but here in the F1 generation I already know that all are same, so I'm just writing a 1 for easy understanding. That is a round and the yellow. Okay, clear? Remember, જો અહીંયા gametes અલગ ફોર્મ થતા હોય તો તમારે બધી 16 possibility બતાવવી જરૂરી છે. You have to show all these 16 possibilities, but here the gametes are same. Even here also the gametes are same. So just we are taking a 1-1 representative because in all the cases they are forming the same round and yellow seeds. So all the plants they are produced in the first generation in dihybrid experiment are round and yellow, and this is a genotype of this plant. Okay, clear? Now they are starting to form the F2 generation. F2. Okay, so I am showing you how to form a F2 generation. So this is a F1 generation. I am writing over here. So which F1 generation we have obtained? We have obtained capital R, small r, capital Y, small y. Now this F1 generation has been self-crossed. Self-crossed means it is crossed with a same variety of plant from the same generation. So same variety, capital R, capital Y, small y, it's crossed with the same variety. This is called as self-cross. So here a self-cross is done. I am writing over here, this is F1 generation. In a F1 generation we are doing a self-cross. Okay. Now first of all, you know that we have to form a gametes. So I am first showing you how to form a gametes. See, I have already shown you how to form a gametes. So the first gamete, I am writing, these are the gametes. Okay, I am showing you first gamete, capital R, capital Y. See here all the gametes will not be same. I am showing you capital R, capital Y. Second, going across, capital R, small y. So here you can see the gametes are changing, capital R, small y. Now see, take this R, small r, capital Y, small r, capital Y. And the fourth one is small r, small y, small r, small y. These 4 gametes are produced of this plant. Okay? Similarly, which are the gametes? Same gametes? Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y, small r, small y. So these are the gametes. Now consider that this is a one plant and this is the another plant. Okay, one is a male, other one is a female. Okay, so these are the gametes. Okay, and these are the gametes. These are the gametes of female plant and these are the gametes of a male plant. Okay, now here the gametes are not same. You can see all these 4 gametes are different. So I have to show you the all 16 possibilities. Okay, for your easy understanding, I am drawing a square. See, I am drawing a square for your easy understanding. Clear? How many square I have to draw? I am explaining with a Punnett square method. So this is a 4 gametes, 4 gametes of a female, and these are the 4 gametes of a male. So how much gametes I have to show? I have to show how much square I have to show? 4 4s are 16. So I have to show 16 squares. So I am just drawing the 16 square. Okay, clear? 16 square. 4 4s are 16. On one side I will write the female gamete, on the another side I will write the male gamete. I am just transferring on one side, because you know that in a Punnett square method, on the one side we are writing the female gamete, on the another side we are writing the male gamete. You can also do a vice versa, the result will be the same. There is no difference. Okay, but here I am just transferring this gamete over here one by one. Okay, so transfer this capital R, capital Y. This is the first one. Next, capital R, small y. Second one. Third one, small r, capital Y. Fourth one, small r, small y. Similarly, we have transferred, so I am rubbing it out. Okay, I will rub down. These are the gametes which are formed. Okay, now I am just transferring to a 1 1 square. 1, 2, 3, 4. I am distributing. So I am also rubbing this down. So these are the male gametes. Okay, so I am writing over here. Here the first is capital R, capital Y. Second one is capital R, small y. Next is small r, capital Y. And the fourth one is small r, small y. On one side it is a female gamete, on the other side it's a male gamete. Clear? Now you know that in a box what you have to write. You have to match the respective column and the row. This is a row and this is a column. You have to match in the respective box. So I am just writing down. Okay? See here. Mix this. Then it becomes capital R, capital R, capital Y, capital Y. Clear? Now mix this. One and this. Capital R, capital R, capital Y, small y. See here, it's a small y. Okay, clear? Now mix this two. Capital R, small r, capital Y, capital Y. Clear? Same thing opens over here. Row and column. Capital R, small r, capital Y, small y. Clear? Do the same way, like this. This row, this column. So here it's a capital R, small y, capital Y. But here, first of all, we are always writing the capital letter to avoid the mistake. To avoid the mistake, always remember, write down the first capital letter. So its phenotype will be easily expressed. So capital Y, small y. Clear? Now go in this row, this column. So here it's a capital R, small y, small y. Now go next. Here capital R, small r, capital R, small r, capital Y, small y. Next go in this direction. Capital R, small y, small y. Clear? Do the same. Capital R, small r. I am writing the capital letter first. Okay? Capital Y, capital Y. Next, capital R, small r, capital Y, small y. Okay? Next, small r, capital Y, capital Y. Say, small r, capital Y, small y. Okay? Next go, carry on. Say, capital R, capital Y, small y. Now go in this direction. Capital R, small y, small y. Like this row, this column. Small r, capital Y, small y. Now go in this. Small r, small y, small y. Okay, and thus you can get a 16th possibility in the next generation, and this generation is F2 generation. Okay, so this is F2 generations you can obtain. You can obtain this F2 generation. Okay, now you have to write it's a phenotype. What is its characteristic? Okay, you have to write it down. Okay, so I'm just writing down it's a phenotype. See, capital R, you know that I am repeating. Capital R is a round, small r is a wrinkle. Capital Y is yellow, small y is green. Okay, I am writing here for your reference. Writing over here for your reference, you can easily. Capital R નો meaning શું છે? Round. Small r નો meaning શું છે? Wrinkle. What is the meaning of capital Y? The capital Y means yellow. Small y means green. You know that dominant character will express, and these are the dominant character. From capital R and small r, this is the dominant capital R. From capital Y and small y, the capital Y is dominant. So જ્યાં સુધી dominant present હશે ત્યાં સુધી dominant will only express, and the recessive will only express, means small r and small y will only express if dominant is absent. એટલે capital R and capital Y is absent, then the recessive character will express. Now write down what is its a phenotype. See, this is round and yellow, so I am writing over here, this is round and yellow. Round and yellow. Okay, this is also again round and yellow. This one is round and yellow, because here it's a round and this one is a yellow. Here also it's a round and yellow. It's a capital R is round, capital Y is yellow. Here you see again round and yellow. Here round. Now see here, there is no capital Y. There are both are small y. So it is a round and small y no meaning ખબર છે, it's a green. બરાબર છે? This small y is a recessive character that will only express when dominant is absent. So here capital Y is absent, so it will express green. Now see, this is round and yellow. Here capital R is present, capital Y is present, so it's a round and yellow. Here round, capital R is present and capital Y absent છે એટલે it's a green. Round and green. Clear? Okay. Next, see here, this is round and yellow. This one is again round and yellow. You can also write down below, but due to a less availability of space, I am writing side by with small letters. But you can make a big table. Small r, right? See here. Here capital R is absent. Both are small r. That means they are recessive character. Here dominant is absent, so the recessive will express. So what is the meaning of small r? It is wrinkled and yellow. Next, here it's a wrinkle and yellow. Now go in this way. Here round and yellow. This one is round, capital Y is absent, so both are small y, so it's a green. Here capital R is absent, so it is a wrinkle and yellow. And this is wrinkle because capital R is absent, and green. Clear? Okay. So now count down. Count. How many are there? Okay. How many? Just count it down. So I'm writing over here. Writing. I'm just writing down this F1 generation, what we have produced. So first of all, counter down. Which are, how many round and yellow are there? So I am writing how many round and yellow. Clear? Just we are counting. So this is first. I am making a square, a star. So you can recognize. Second, third, fourth, fifth. This is not round yellow, this is round yellow. Sixth, seventh, eighth, nine. So there are round and yellow. How many round and yellow? 9. 9 are round and yellow. Clear? Okay. Now we count the another one. It is round and green. Round and green. Okay. So we are counting round and green. How many round and green are there? Okay. So we are just counting round and green. So I am just making a triangle so you can identify. This is round and green. This is again the second one, round and green. Okay. Next, this is the third one, which is a round and green. Okay. So how many round and green are there? These are 3. We go for the third possibility. It is wrinkled and yellow. Wrinkled and yellow. Okay. So we count which are the wrinkled and yellow. I am making a square for you easy understanding. This is the first one, which is wrinkled yellow. This is the second one, which is a wrinkled yellow. This is the third one, which is a wrinkled yellow. So how many? The wrinkled yellow are again 3. And last possibility, that what we are discussing, that is a wrinkled green. So here, how many wrinkled and green? Just we are counting out. There is only one left. Here there is only one left. So this is the wrinkled and green. How much it is? 1. Okay. So 9:3:3:3:1. This ratio is obtained in F2 generation. In which generation? F2 generation. Which kind of ratio it is? It is a phenotypic ratio. So in a dihybrid experiment, the phenotypic ratio in the F2 generation is 9:3:3:1. 9 are the round and yellow, 3 are the round and green, 3 are the wrinkled and yellow, the 1 is wrinkled and green. This possibility are there. If you make the total, 9 plus 3 12, 12 plus 3 15, 15 plus 1 16, so total 16 possibility we have obtained. This is called as dihybrid experiment. What it is called as dihybrid experiment. I again just revised what the Mendel has done in a dihybrid experiment or inheritance of a 2 gene. First he took the round and yellow parent, he crossed it with wrinkled and the green. In the first generation, all the plants obtained are the round and yellow with this genotype. Now he self-crossed this F1 generation. So he got this F2 generation. You know that how to do this. On one side male gamete, other side female gamete. You have to mix it. And then you count. Then how many round yellow? 9. How many round green? 3. How many wrinkled yellow? 3. How many wrinkled green? 1. Okay. So this is what happens in the dihybrid experiment or inheritance of a 2 gene. Okay. So this is the phenotypic ratio has been obtained. Now from the homework you have to find out what is its genotypic ratio. Phenotypic ratio तो मैं तमने count करीने आपी दीदो. Now you have to count the what is the genotypic ratio. Means you have to count how many capital R, capital Y, capital Y is present. How many capital R, capital Y, small y is present. Okay, just count it. Similarly, बराबर છે બધા તમારે count કરીને લખવાના. Like suppose genotypic ratio capital R, capital Y, how much they are counted. Second possibility capital R, capital Y, small y. How many? This is the third one. Capital R, small r, capital Y, how many? बराबर છે? Clear? So you want to find out what is the genotypic ratio. By writing this, that you have to do in a homework. You just have to count it. Okay? So this is called as dihybrid experiment or the inheritance of a 2 gene. Okay? Now, what we have understood from this experiment. See here. To understand what we have learned in this, first of all, you should also know that monohybrid experiment. Like in monohybrid experiment, the tall plant is crossed with a dwarf plant. In the F2 generation, either you get a ratio 3:1. So 3 tall, 1 dwarf. This is the ratio that is obtained in the F2 generation. This is the parent generation in monohybrid experiment. એટલે parent તરીકે tall હતા અને dwarf હતા, તો F2 જનરેશનમાં બી ટોલ અને ડ્વાર્ફ જ આવશે. એ સિવાયની કોઈ પોસિબિલિટી છે નહીં. there is no other possibility. ક્યાં ટોલ હોય ક્યાં ડ્વાર્ફ. પણ અહીંયા જુઓ, અહીંયા as a parent generation what we have taken, round and yellow. First parent is round and yellow, capital R, capital R, capital Y, capital Y. That has been crossed with wrinkled and green. So that is small r, small y, small y. હવે મોનોહાઇબ્રિડ પ્રમાણે ગણીએ તો F2 જનરેશનમાં F2 બરાબર, એ F2 જનરેશન પ્લાન્ટ્સ કયા આવવા જોઈએ? ખાલી રાઉન્ડ યલો અને wrinkle green. બરાબર, રેશિયો ભલે ગમે તે હોય, round yellow and wrinkle green, but here you can also find some other possibility. A round green, a new combination is formed, a round green, and even you can also found the another possibility, that is a wrinkled yellow. That is also આ તો બે પેરેન્ટ છે જ, રાઉન્ડ યલો પેરેન્ટ છે, wrinkle green બી પેરેન્ટ છે, but અહીંયા પેરેન્ટ કરતા અલગ પ્લાન્ટ also have been developed or generated. That is a round green and wrinkled yellow plants also have been developed or generated in this ratio. So that says that here the assortment, separation of gamete formation, they occurs independently, and based on that the Mendel has proposed one more law, that is called as law of independent assortment. What it is called as? Law of independent assortment. What the law of independent assortment says? See here what the law of independent assortment says. Here capital R, small r, capital Y, small y. These are the gametes. Sorry, this is the F1 generation. One of the plant in F1 generation. Capital R, small r, capital Y, small y. Now where it is crossed with same self cross. So what happens? The gametes usually in a parent cover to round and yellow. But here the round gene or the gene for a round, that is capital R, also goes with a small y. Capital R also goes with a small y. That means the assortment that occurs independently. There is no necessity कि capital R जोड़े हमेशा always capital Y जावो જોઈએ. There is also a possibility that capital R ने जोड़े small y पण આવે. So that means the assortment occurs independently. એવી જ રીતે સેમ સ્મોલ r જોડે સ્મોલ y જવો નેસેસરી નથી. સ્મોલ r જોડે કેપિટલ y બી આવી શકે છે. So this is the third gamete what we have found, and this is the fourth gamete. Okay, this is called as law of independent assortment. Law of independent assortment. That means assortment occurs independently. The capital R can also go with small y, though it is not present in a parent. Okay, so this is a law which was proposed by the Mendel based on the dihybrid experiment or the inheritance of a 2 gene. Like I will also explain you with some more example. This is an example in a pea plant, because you know that Mendel has performed his all experiment in the garden pea plant, that is a Pisum sativa male. Okay, but you can see I will give you some hypothetical example. Suppose one boy with black hair and curly hair that marries with a girl which has a white hair, or you can say instead of white I am writing a brown hair and straight hair. Here the boy is black hair with a curly, the girl is with a brown hair and the straight hair. Now this is a parent generation. So if you see the F2 generation 2, in the generation 2 there are chances that a human is going to be produced which might have a black hair and they have a black and the straight hair, and even a possibility is that of production of human there is a which has brown hair with a curly hair, like what happened over here. So this happens, that means that says that the assortment it occurs independently. Okay, so I hope so you understood what is a dihybrid experiment. Here the formation is gamete is very important. If you know this formation of gamete, then the other thing is very easy. Okay, in the homework I have already told you, find out the genotypic ratio of this F2 generation. Phenotypic ratio I have already told you, 9:3:3:1. Try to find the genotypic ratio. If you try, then it is a lot beneficial to you. Okay, so students, learn it, keep learning, keep watching, like and subscribe this channel. Thank you for watching.